Today, we are going to crack one of the oldest mysteries in all of science. It's a simple question with a fascinating answer. How are traits passed down from a parent to their child? Let's get right into it. For the longest time, this question completely stumped the greatest minds. The popular idea was that traits just kind of blended together, like mixing two colors of paint. But that didn't really make sense, did it? How could a trait like blue eyes suddenly reappear after skipping a generation? We needed a detective to solve this case. And that detective, it turns out, was an Austrian monk named Gregor Mendel. Right there in a quiet monastery garden, he was about to uncover the rules that would become the foundation for the entire field of genetics. Now, this wasn't some flash-in-the-pan discovery. Mendel's work kicked off in 1853, and it took 8 long years of incredibly careful and patient experimentation. He published his amazing findings in 1866, but believe it or not, it took decades for the rest of the world to really catch on and realize just how important his work was. A huge part of Mendel's success really came down to one brilliant choice. To solve the big mystery of inheritance, he knew he needed the absolute perfect subject to study, and he found it in the common garden pea plant. So, why peas? What's so special about them? Well, they were kind of the perfect lab partner. They grow super fast, so he could study many generations in a short time. They also produce tons of offspring, which gave him a lot of data to work with, making his results more reliable. And here's the real kicker. They had a bunch of simple traits that came in only 2 clear forms, things like tall or short, round or wrinkled. There was no confusing in between. See, Mendel's real genius was in keeping it simple. Instead of getting overwhelmed, he decided to track just one single trait at a time. This is what we call a monohybrid cross. So we're going to follow along with his very first experiment, and we're going to focus on one simple trait, plant height. Okay, so he started with what's called the parental, or P generation. To make sure his starting point was totally clean, he used what are called purebred plants. That means he had a tall plant that, when it self-pollinated, only ever made tall offspring, and he had a short plant that only ever made short offspring. Then he crossed them. So, what do you think happened? Did he get a bunch of medium-sized plants? A mix of tall and short? Well, the actual result was the first huge clue in this entire mystery, and honestly, nobody saw it coming. Get this. Every single one of the new plants in this first, or F1 generation, was tall. Not a single short one, not one medium one. All tall. It was as if the short trait had just vanished into thin air. This flew in the face of that whole blending idea. And this right here is the million-dollar question that led Mendel to his big breakthrough. The trait couldn't have just disappeared forever, right? It had to be hiding somewhere. But how is that even possible? Mendel figured that for every trait, an organism gets 2 factors, one from each parent. Today, we don't call them factors. We call them alleles. Think of them as just different versions of a gene. And here's his insight. He realized one of these alleles could completely hide or mask the effect of the other one. He called the trait that shows up the dominant allele, and to keep things straight, we represent it with an uppercase letter, so uppercase T for tall. The trait that gets hidden he called recessive, and we represent that with a lowercase letter, so lowercase t for short. This means that every single one of those F1 plants had the exact same genetic makeup, or what we call a genotype. They got one dominant allele from the tall parent, that's uppercase T, and one recessive allele from the short parent, that's lowercase t. So even though they looked tall, they were all secretly carrying the instructions for being short. So what's a good scientist to do next? Well, Mendel took 2 of these hybrid tall plants from the F1 generation, and he crossed them with each other. He just had to know, could that hidden short trait ever make a comeback? So for this second cross, the one that makes the F2 generation, he was crossing a plant with the genotype uppercase T, lowercase t, with another plant that was also uppercase T, lowercase t. This meant that each parent could now pass on either the tall allele or the short allele. The possibilities were opening up. Now, to see all the possible outcomes, we can use a really simple but incredibly powerful tool. It's called a Punnett square. It's basically just a little grid that helps us map out and predict the probability of what the next generation's genes will look like. Okay, let's fill this out together. We'll put the alleles from one parent across the top, so uppercase T and lowercase t. And we'll put the alleles from the other parent down the side. Again, uppercase T and lowercase t. Now we just combine them in the boxes. Top left gets an uppercase T from the top and an uppercase T from the side, so that's uppercase T, uppercase T. Top right gets uppercase T, lowercase t. Bottom left also gets uppercase T, lowercase t. And finally, the bottom right box gets lowercase t, lowercase t. So now if you look at our results, you can see a really clear pattern in the genotypes. We've got one box with uppercase T, uppercase t. We've got two boxes with uppercase T, lowercase t, and we have one box with lowercase t, lowercase t. This gives us a really precise genotypic ratio of 1:2:1. But what do the plants actually look like? That's the phenotype. Well, the uppercase T plant is obviously tall, and the two uppercase T, lowercase t plants are also tall. Remember, because that uppercase T is dominant. It's only the lowercase t plant that ends up being short. And right there, that gives us Mendel's famous 3:1 phenotypic ratio. For every 3 tall plants, that hidden short trait showed up once. It was back. If you think about it in terms of probability, it's pretty cool. There's a 75% chance that an offspring plant will be tall and a 25% chance it'll be short. That trait didn't just reappear out of nowhere. It came back in a perfectly predictable mathematical way. Amazing. This predictable pattern, this reappearance of the recessive trait, led Mendel to his first huge conclusion. It was a fundamental rule of genetics that finally solved the mystery of how traits get passed on without just blending into nothing. He called it the Law of Segregation. Now, that sounds complicated, but all it really says is that when an organism creates its reproductive cells, like sperm or eggs, its 2 alleles for a trait pull apart, or segregate. That way, each new cell only gets one of the 2 copies. And that's why a hybrid parent can pass on either its dominant trait or its recessive trait, but never both at the same time to the same offspring. And with that one simple law, you now have an incredibly powerful tool. You can use this exact same logic to predict the results of any simple monohybrid cross. All you gotta do is follow these 5 steps. Identify the alleles, write out the parents' genotypes, figure out the possible gametes, draw your Punnett square, and then just calculate the final ratios. You can do this. And there you have it. The beautiful, elegant logic behind how a single trait is passed from one generation to the next, all figured out in a garden over 150 years ago. But of course, that just leads to the next big question, doesn't it? What happens when you try to track 2 different traits at the same time, like not just height, but height and seed color? Well, that's a story for another explainer.